Hey everybody, welcome to I think the first mini lecture for Module 5, and here we will pick up where we left off in Module 4 and talk about gene mutations. So what are mutations? Mutations are changes to nucleotides in a DNA or mRNA sequence. They occur because of damage to DNA. There are two types of mutations that cause DNA damage: induced mutations and spontaneous mutations. Induced mutations generally come from environmental exposure. This could be caused by chemicals, like the toxic chemicals in cigarettes or chemical vaping solutions. It could be caused by UV rays, like those from the sun, or X-ray exposure, which is why we have to cover our bodies with those heavy drapes when we are giving or getting X-rays. We hear about another example on radio and TV ads about asbestos causing mesothelioma. Asbestos is a cancer-causing agent that induces mutations, so it's another example of chemical exposure. In this figure, we see a UV ray inducing a double-stranded DNA break. The UV literally snaps the DNA strand in half, preventing any kind of replication, transcription, or translation. In this example of induced DNA damage, we see that a cytosine, seen over here, is deaminated, which means it loses its amino group. And this amino group is what gives the cytosine its identity from the other base pairs. Now, because of the deamination, the cytosine actually looks like a uracil. This will interfere with replication because we know that DNA is not even supposed to carry a uracil. So the transcription machinery will make a big mistake and pair this with an A, which is uracil's partner, instead of the G, which would have been if it was still cytosine. So the replicated DNA strand sequence will change. Later, we'll talk about repair mechanisms that are constantly battling DNA damage, because this kind of stuff happens all the time in our DNA, but our bodies are really capable of catching it before it becomes an issue. The second class of DNA damage are spontaneous mutations. Spontaneous mutations don't need external or environmental or chemical causes. They just happen randomly. Spontaneous mutations, though, aren't always bad. These are the same kind of mutations we were looking at when we talked about genetic drift. So the mutations that change, or even the pocket mouse example. So mutations that change the fur from brown to black, they just happen randomly and spontaneously, but we can see from our pocket mouse lab that they conferred some enhancements based on where the mice lived. But in this example on this slide, we see that during DNA replication, the A on the bottom strand is copied as a C instead of a T. So the daughter cell strand contains a new DNA sequence. This wild type will replicate normally, and all of its daughter cells will go on as expected. But the mutated one now, when the bottom strand is replicated, we see that the sequence is now TGCG instead of TACG. This will have an effect on transcription and translation going forward. Either way, these single base pair changes can completely change the DNA sequence, no matter if it's an induced or a spontaneous mutation. We saw in Module 4 that individual base pairs are very important in determining which amino acid will be presented, and we know that amino acids are the limiting factor in determining which protein will be produced. So a single base pair change can mean the difference between a protein being produced or not. If we follow the schematic in this slide, we see that some mutagenesis event targeted the C here in this top strand of the DNA sequence. And although replication occurs normally on the bottom template strand, the top template strand actually accidentally pairs an A with the C instead of pairing a G with the C. When we look in the last panel, we see that the DNA sequences are different now in the mutated strand. The top strand is repaired by normal DNA repair mechanisms, which we'll talk about later, but the bottom strand still contains the mutation, and this mutation will be passed on to its daughter cells. Now it's your turn to practice. Look at the two sequences below. We have a wild type sequence, ATG, and a sequence that has been mutated and now reads ACG. First, transcribe these 3 DNA base pairs into mRNA. Secondly, translate the mRNA sequences to an amino acid. And then look closely and see if this mutation affects translation to amino acids. Pause now and take these 3 steps. The next slide contains a solution, but give it a try first. When we transcribe the strands to mRNA, we see that the ATG transcribes to a UAC, which translates to a tyrosine amino acid. But on the mutated strand, the ACG now transcribes to a UGC, and in translation, that becomes a cysteine. So the effect on translation is that 2 different amino acids have been produced. Is that what you got when you tried it? Okay, we'll continue on to the next activity in the module. Hope this is a good primer and you feel a little bit more knowledgeable about what DNA mutations are.